lab that relate to the molecular basis of cardiovascular disease and various strategies for correcting those disorders. So let's try to advance my slides. Well, so my slides, there we go. These are my disclosures. Okay. My lab, as you mentioned, has studied the three major muscle cell types: skeletal muscle, cardiac muscle, and smooth muscle, with the goal of identifying the gene regulatory networks that control the formation and function of these different muscle cell types. And along the way, many of those molecules and mechanisms that we've uncovered have been found to play critical roles in disorders of each of these muscle tissues. Today, I'm going to talk to you about two projects from my lab. I'll begin by telling you about our deployment of a strategy called myoediting for correcting muscle and cardiovascular disorders at the genetic level. And then if time permits, I'll talk to you about a new project that focuses on the molecular basis of cardiovascular gene expression through the formation of molecular condensates. Muscle tissues are plagued by literally hundreds of genetic disorders. These are devastating disorders that have defied all therapies to date, and importantly, there are no cures for any. So this represents a real frontier in molecular biology and medicine, and a great area for future investigation for trainees. One of the most devastating and defiant of all muscle disorders is Duchenne muscular dystrophy, abbreviated DMD. This disease was first identified by the French neurologist Guillaume Duchenne in 1860. Patients diagnosed with Duchenne first appear with walking problems at around the age of 2, which rapidly progress to the loss of ambulation, such that these patients, who are almost all boys, as we'll discuss, become confined to wheelchairs by the early teens and ultimately lose the use of their arms, and they become confined to a ventilator because the diaphragm disintegrates, and they ultimately succumb, sadly, to heart failure in the mid-20s. So far, no patient diagnosed with Duchenne muscular dystrophy has defied these pathological consequences of the disease. And so this is a huge area of unmet medical need and opportunity. The disease is caused by mutations in the dystrophin gene, the largest gene in the human genome. The dystrophin gene spans 3.5 megabases of DNA and encompasses 79 exons. To give you a sense of how large 3.5 megabases of DNA actually is, it's estimated that it takes at least 12 or more hours for RNA polymerase to traverse this gene one time and make a single copy of the RNA. The gene is located on the X chromosome, hence a mutation in the single copy of this gene in boys inevitably leads to the disorder. And there have been thousands of mutations identified in this gene, which represents one of the major challenges. The dystrophin gene encodes the dystrophin protein schematized on the lower left. And you can think about this as a molecular scaffold or a shock absorber for muscle. It connects the cell surface adhesion complex known as the dystroglycan complex with the underlying actin cytoskeleton, and its presence is essential to maintain the integrity of muscle membranes during each cycle of contraction and relaxation. The structure of the dystrophin protein is schematized here at the top. You can think about this as a molecular shock absorber, with the two ends being essential to anchor it in place at the actin cytoskeleton and the dystroglycan complex. The central rod domain contains 24 redundant rods, analogous to coils on a spring. And you can see here the color-coded exons, the 79 exons that encode each of these proteins, portions of the protein. And you can see how these exons splice together seamlessly like pieces of a jigsaw puzzle to form the ultimate intact mRNA. Now, although there are thousands of mutations that have been identified in patients worldwide, they tend to cluster into these 10 red exons. These are known as hotspot mutations, and they account for anywhere from 60 to 80% of the mutations that have been identified. In looking at these exons, you can see why a deletion of any of these exons would lead to Duchenne, because these exons are asymmetric, such that if you remove one of these exons, the two flanking exons are out of frame, and that imposes a premature termination codon that prevents the translation of the essential C terminus. This has led to the concept of exon skipping, in which one might skip an out-of-frame exon and restore the continuity of the open reading frame by connecting an upstream exon to an in-frame downstream exon. And the rationale for this approach is based on a relatively mild muscle disorder called Becker's muscular dystrophy, or BMD. These patients harbor in-frame deletions within the rod domain, but they contain a sufficient number of rod domains to enable them to maintain ambulation, often into the 6th decade of life. So this has led to the concept of converting DMD into BMD as a strategy for restoration of dystrophin function in these patients. We had thought deeply about Duchenne for many years, but never felt that we had a way to appropriately address this disorder until the advent of CRISPR. And with the advent of CRISPR, we decided to test whether we could correct mutations that cause this disease by correcting the actual underlying genetic defect. Now, in our first version of CRISPR gene therapy, we used a Cas9 nuclease, schematized here, which, as you all know, can be delivered to any sequence in the genome with a single guide RNA. And when an appropriate PAM sequence is located nearby, this will engineer a double-stranded DNA break. The double-strand break can be repaired by either of two mechanisms. Homologous recombination involving a donor template can lead to perfect replacement, but this pathway does not exist in quiescent cells, and so we knew we could not deploy homologous recombination to correct these mutations. So we relied on the second pathway, the non-homologous end-joining pathway, in which the double-strand DNA break can be repaired by the endogenous DNA repair machinery, which in some cases will leave insertions or deletions, so-called indels, at the break site. But if an indel inserts or removes an appropriate number of nucleotides, it can restore the continuity of the reading frame between the upstream and the downstream exons that would otherwise be out of frame. So why did we think CRISPR would be an appropriate therapy ultimately for Duchenne? Well, there were many reasons for this decision. One is that the dystrophin gene, as I've told you, is simply far too large to replace by traditional gene therapy. Secondly, the modular gene structure of the dystrophin gene allows exon skipping to restore the dystrophin protein, converting DMD into BMD. Finally, normal levels of dystrophin expression are thought to not be necessary for benefit, and this is a very important point. It's been estimated that as little as 15% of normal dystrophin levels could be curative for this disorder. And because many muscle tissues, and particularly the heart, which is the ultimate cause of death in these patients, are permanent tissues, we thought that it might be possible for a one-time treatment to correct this disorder. So this is how we began. We set up a muscular dystrophy clinic here at UT Southwestern in Dallas. We can take blood samples from patients who have mutations in the dystrophin gene. We convert them into iPS cells, which can then be converted into muscle cells, either skeletal muscle or cardiac. Then we can optimize gene editing approaches in these cells. This is what they look like. So here's a blood sample from one of our first patients, converted into beating cardiomyocytes, and then we can also incorporate those into what's known as engineered human heart muscle and study their physiology and function. So this provides us with a reliable preclinical humanized model for testing and optimizing gene editing therapies in vitro before going into in vivo models. Here's an example of how this strategy works. So at the top, you can see a scheme of the dystrophin protein. And beneath that, you can see exons 42 through 46 of the dystrophin gene, which encode a region of the rod domain. This was a patient. He was lacking exon 44 of the dystrophin gene. You can see why this boy had Duchenne, because exon 43 will splice to the next exon in line, which is exon 45, but that imposes a premature termination codon because it's out of frame with the preceding exon. So we screened for guide RNAs with Cas9 or other Cas endonucleases to ultimately identify optimized guide RNAs that can correct this mutation in either of two ways. If the guide RNA inserts an indel, an insertion or deletion in exon 45, and it contains the appropriate number of nucleotides, it can reframe exon 45 and put it back into a contiguous open reading frame with the preceding exon 43. Alternatively, if we direct Cas9 to delete or mutate the splice acceptor sequence at the 5' end of exon 45, it will render that exon invisible to the splicing machinery, and exon 43 will splice to the next exon in line, which is 46 in this case, and fortuitously those two fit back together so they reconstitute a functional dystrophin protein. This is how it works. These are human iPS-derived cardiomyocytes from a patient. Here's the dystrophin protein on a Western blot. Here's this patient's cells. They make no dystrophin, and when we correct by single-cut CRISPR, we can restore dystrophin. Importantly, when one corrects the mutation in these patient-derived cells, and we believe ultimately in patients, we hope, that the corrected gene is expressed at the right time, the right place, and the right level as the endogenous gene because the correction occurs within the endogenous locus. This shows the cardiomyocytes from a normal patient here with dystrophin in red. Here's from this patient lacking dystrophin, and then following single cut correction, dystrophin is expressed back at normal level, and this also normalizes other physiological functions of myocytes, including calcium cycling, as you can see here. So in our version, you could call this version 1.0, we engineered an SP-Cas9 endonuclease under control of a muscle-specific promoter to restrict its expression to cardiac and skeletal muscle, and we used an adeno-associated virus 9, which has tropism for muscle and heart, to deliver this to mice. Because this version of Cas9 is too large to be incorporated into AAV, together with the guide RNAs, we used a second AAV to deliver the guide RNA under control of appropriate promoters into mice. This shows the results. Along the top, you can see dystrophin staining in the submembranous region of skeletal muscle, diaphragm, and cardiac muscle. In the middle row, you can see this mouse model of Duchenne lacking dystrophin. And here you can see the correction of this mutation and the restoration of dystrophin protein at approximately the normal level and the normal subcellular distribution in these animals. And we went on and work that's been published to show that the restoration of dystrophin in these animals restores normal muscle integrity, physiology, function, and gene expression. We wanted to test whether we could deploy this method to larger animals with Duchenne. We searched the world over and identified a family of dogs that maintained at the Royal Veterinary College in London that fortuitously harbored the same mutation, which was the first one that we had corrected in my neighbor here in Dallas in his cells. So we went to the Royal Veterinary College and we treated these dogs with AAV9 of the structure that I showed you on the previous slide, and I'll show you a video of the correction of these dogs. So here you can see, first you'll see 3 dogs in this series of videos, all from the same litter. This is a normal puppy. I'll tell you, they're extremely cute. Now in the next video, you'll see his brother from the same litter who has Duchenne, and you'll see that his muscles are severely atrophied and his ambulation is impaired. So this is a dog at the early stages of Duchenne, but I think you can even see at this stage that this dog has difficulties ambulating. And now you'll see his brother, who also has Duchenne, who we treated with the virus with a single dose. This is 8 weeks after that single administration of the gene editing therapy. And I think you can readily see this is a happy dog with normal muscle mass, normal muscle function, ambulation. He can run, jump, and is functionally and physiologically identical or comparable to his wild type littermate. So based on these data, we feel quite optimistic about the potential of this therapy, and we've gone on to try to optimize it further, which I'll talk about in the next slides. So the first version of CRISPR that I described was single-cut CRISPR, which we showed was safe and effective in mice, human cells, and dogs. But it does impose a double-stranded DNA break, and so we wanted to test whether we could correct Duchenne by not making a double-stranded DNA break. And with the advent of base editing, as pioneered by David Liu at Harvard, we felt there might be a way to correct this disorder using this strategy. Now, in this strategy, which I think is familiar to almost everyone on this call, again, it uses a Cas9. In this case, it's a crippled Cas9 that doesn't make a double-strand break, and it's fused to a deaminase domain that within an editing window dictated by the guide RNA can make single nucleotide changes. In cytosine base editing, a CG base pair can be converted to a TA base pair, and in adenine base editing, an AT base pair can be converted to a GC base pair. So we ask ourselves, how could we deploy a base editor to change a single nucleotide in a genome and correct a large deletion in the dystrophin gene? And we realized that every exon is preceded by a canonical dinucleotide splice acceptor sequence and is followed by a canonical dinucleotide splice donor sequence, which fortuitously contain the target sequences for A or C base editing. So we reasoned if we could edit either the splice acceptor or the splice donor, we would render the out-of-frame exon invisible to the splicing machinery, and then the preceding exon would splice automatically to the following exon, in this case shown schematically as exons 1 skipping 2 to exon 3. And so here's how it works. We engineered mice with humanized dystrophin mutations surrounding exon 51. This is the most commonly deleted region in patients with Duchenne. Here's the wild-type mice. Here's the mice lacking exon 51. You can see they're unable to make normal levels of dystrophin because these two exons are out of frame. If we base edit either the splice donor, in this case, at the 3' end of exon 50, but it also works if you edit the splice acceptor at the 5' end of exon 50, the exon just is bypassed by the splicing machinery, which will then splice 49 to 52. Fortuitously, they come back in frame, and there you see the normal level of dystrophin restoration. So these are the most commonly deleted exons in boys with Duchenne, and this shows the patient percentages represented by each of these deletions. To optimize further humanized gene editing, we generated humanized mice with the human genome sequence within each of these regions, and we've optimized base editing for all of the targets that I've shown you here, in either splice donors or splice acceptors, all of which can restore dystrophin expression. So we've wanted then to go on and try to optimize this further because the base editors are too large to fit into a single AAV, which necessitated the use of a split intine system to reconstitute the functional base editing system. But to avoid high doses of AAV and to further optimize this method, Andy Pian and Hui Li and others in our lab have optimized base editing. They did so by screening 9 miniaturized A base editors along with approximately 87 guide RNA combinations to optimize editing of these out-of-frame exons, all of which represent the most commonly deleted exons in boys with Duchenne. And the ultimate optimized mini we call it an all-in-one base editor is shown here. It's an N-slug Cas9 fused to an ABE8E for the aficionados in the audience. And it contains a muscle-specific promoter and as well as a guide RNA cassette. We've used this, in this case, I'll show you one example, to correct an exon 44 deletion by targeting a splice acceptor sequence at the out-of-frame exon 45. And we've done this by single intravenous injection through retroorbital injection into mice, adult mice at a dose of 5 × 10^13. And I'll come back to that dose in a moment. And here you can see the results. Here are saline injected animals. They're devoid of dystrophin in these 3 representative muscle tissues. And following treatment with a single all-in-one base editor, we can restore dystrophin to reasonable levels of expression in all these tissues. And for these studies, which I won't have time to go into in detail, we've used two versions of an AAV that have specific tropism to muscle and heart, known as Myo4A AAV. And we've also coupled this with a liver-off system developed by Luc Vandenberg's group to further narrow the target specificity of this AAV. And here you can see the levels of expression delivering over 50% of the normal level of dystrophin protein in the heart and approximately 20% at this early time point in these other muscle tissues, diaphragm and tibialis anterior. So I'd like to speak for a moment about the structure of these edited dystrophin proteins relative to other mini dystrophins that are currently in therapeutic use. So the full-length dystrophin protein is 3,684 amino acids in length with 24 rod domains, as you can see here. When we correct an exon 50 deletion by either base editing or single-cut CRISPR, it restores 3,606 amino acids of the protein, almost full length, and functionally, we believe, virtually equivalent to normal. This is in contrast to Becker's muscular dystrophy, BMD. This is the smallest, to my knowledge, dystrophin protein that's been identified in patients with BMD that retained ambulation. You can see this patient retained a subset, approximately 9 rod domains, but is missing a very large portion of the central region. In contrast, there has been many therapeutic trials now from microdystrophins from Sarepta Therapeutics and other companies. This is the currently used therapeutic from Sarepta. You can see its size relative to BMD, and there has not yet been a Becker patient that harbors a dystrophin protein of this particular size. And there have been issues associated with the Sarepta trials due to high-dose AAV, which we and others are currently working to overcome. So an important question from these studies and the work of many other groups that are involved in this field is, when would one want to intervene? We believe the earlier the better. Once muscle is severely damaged, there's no substrate left to correct, so we really are pushing for early intervention at the time of genetic diagnosis. Another important issue is safety. Toxicity of AAV is an issue of high concern. Off-target editing is often mentioned as a concern, although we have not seen significant off-target editing in any of our studies, either short or long-term, and I haven't had time to go into that data today. And lastly, immune reaction to AAV or to Cas9 is an issue that needs to be confronted and probably will be ameliorated by immunosuppression. Another important issue is the mode of delivery. I've focused so far on viral delivery, but we think that non-viral delivery is the ultimate solution to this disease, and so we're beginning to explore that possibility. In one series of studies that we've carried out in collaboration with Daniel Seagort's lab at UT Southwestern, we've attempted to optimize base editor mRNAs by combining them with guide RNAs and various recipes of lipid nanoparticles, initially to correct Duchenne mutations in iPSC-derived cardiomyocytes. This is one example in which we've used an A base editor to correct, again, an exon 44 deletion by editing the splice acceptor sequence at the 5' end of exon 45. You can see this particular base editing strategy leads to very high efficiency of editing with virtually no bystander editing. And here in vitro, you can see these are the patient-derived cells devoid of dystrophin. And here, following base editing in 3 triplicate cultures, you can see dystrophin protein restored to a level of about 80% of normal. So we're quite enthusiastic about this approach. We've begun to move these lipid nanoparticle base editors in vivo. This shows just one example of an intramuscular injection of that optimized lipid nanoparticle-based base editor, and you can see that it was sufficient to restore dystrophin protein intramuscularly in this animal. But we ultimately believe that systemic delivery through intravenous delivery will be the eventual solution. We're currently screening lipid nanoparticles through a number of strategies to further optimize this approach. I'm just going to summarize this quickly here before moving on. We are screening for endothelial and muscle cell ligands that can be used to couple LNPs to targeting moieties that would take them to the tissues of interest. We're varying the lipid composition of various LNPs, and we have a paper that should appear soon describing this together with the Seegwart lab. We're working to conjugate a unique muscle fusogen discovered in our lab several years ago called MyoMaker to LNPs to deliver them specifically to muscle, or to use a monoclonal antibody against MyoMaker to deliver these to muscle tissue. And in our most recent work, which I won't have time to go into today, we're using AI design to develop unique targeting peptides for specific receptors in the target tissues of interest. So I've told you then about some of our work on Duchenne, and we're trying to build on our knowledge of gene editing for muscle to deploy these strategies for some of the many other devastating disorders of muscle tissues. In recent work that was published, we've corrected lamin A myopathies. I encourage you to look at that paper. We've also corrected hypertrophic cardiomyopathies in mice and humanized cells harboring mutations in myosin heavy chain 7 and dilated cardiomyopathies harboring mutations in RBM20. I'd like to spend the next few minutes talking about another cardiovascular disorder, that is aneurysms, which are obviously a very severe represent a very serious cardiovascular disease. And I'll talk to you about one especially aggressive genetic form of aneurysm caused by mutations in the ACTA2 or smooth muscle actin gene. This disorder often leads to thoracic aortic aneurysm and dissection, abbreviated TAAD. The ACTA2 gene, which leads to this mutation, can harbor a particularly pathogenic variant called the R179H mutation. This is extremely severe. It causes multisystemic smooth muscle dysfunction and fatal aneurysms by ages 20 to 30. And it does so by disrupting actin polymerization and smooth muscle contractility. This slide illustrates the dynamic phenotypic switching that occurs between smooth muscle cells, which can transition between a differentiated state, which maintains the contractility of the vasculature and all other smooth muscle containing tissues in the body, and smooth muscle dysfunction, which provides the cellular basis for aneurysm. And the phenotypic plasticity and function of smooth muscle cells is dependent on the state of actin polymerization. When F-actin is polymerized, it maintains smooth muscle contractility, but in the setting of the R179H mutation, actin polymerization is disrupted, and this tilts the balance towards smooth muscle dysfunction, lack of contractility, and aneurysm. We wondered whether it might be possible to deploy a gene editing strategy to correct this disorder. The mutation responsible for this disease is located in exon 6 of the actin genes, shown schematically here. And here you can see the arginine 179 to H mutation, which fortuitously involves a G to an A mutation that we thought would be highly amenable to adenine base editing to revert the mutation back to the wild type guanine. So in screening large numbers of guide RNAs, we settled on one schematized here and combined it with a variant of a base editor. For the aficionados in the audience, this is the base editor that ultimately proved the most efficient. And here you can see the efficiency of base editing of that mutation, upwards of 80% with virtually no off-target or bystander editing at this adjacent adenine nucleotide. So we wanted to know whether base editing could indeed restore the normal physiology and function of these defective smooth muscle cells. And so we set up in vitro assays in human iPS-derived smooth muscle cells harboring this mutation. And here you can see that the proliferation of these mutant cells is enhanced because these cells aren't differentiating correctly. And when we correct them by base editing, it restores proliferation to normal. Similarly, the migratory activity of these mutant smooth muscle cells is enhanced, preventing their normal differentiation. This can be normalized by base editing. And finally, the state of actin polymerization, which is really the molecular underpinning of this phenotype. Actin polymerization is deficient in these mutants and is restored to normal following gene editing. One can also study the contractility of smooth muscle cells in this very visual assay. If one plates smooth muscle cells on a dish, as shown here, they are spontaneously contractile and they contract here into a tight ball. The R179H smooth muscle cells are deficient in their ability to contract, and so they remain as a monolayer of cells here. But when we correct them by single base editing, it restores their contractility, and you can see here they've contracted back into a contractile ball. We wanted to test whether we could deploy this optimized base editing strategy in vivo. So we first screened a number of smooth muscle specific promoters and ultimately found a very optimum smooth muscle promoter that would restrict the expression of the base editor to all smooth muscle cells, both vascular and visceral, but not to non-smooth muscle cells. And we engineered that into AAV. And here you can see the phenotypic consequences of this correction. So on the top, you can see 3 representative tissues that are dependent on smooth muscle cells for their function. Here's the intact structure of the aorta, the bladder, and the kidney. Here are mice that harbor the heterozygous mutation at R179H. You can see this mouse has a severe aneurysm with a thrombus in the atrium. Here you can see the bladder is severely dilated and lacking in contractile smooth muscle cells. And you can see here in the kidney, this animal has severe hydronephrosis due to defects in kidney smooth muscle cells. And here you can see following a single dose of this base editor under control of the smooth muscle specific promoter that the structure, and I won't show you, but the function of all these smooth muscle containing tissues is completely normalized. So what I've told you then so far is that the properties of smooth muscle cells are dictated by actin polymerization, particularly the polymerization of ACTA2, the smooth muscle specific isoform, and that the R179 mutation, which is an insidious mutation seen in patients, leads to smooth muscle dysfunction, aneurysm, and absence of contractility of many smooth muscle containing tissues. I've told you that F-actin polymerization underpins the function of all smooth muscle cells in the body, and that R179H blocks actin polymerization, and hence smooth muscle cells are unable to function appropriately. Beneath the dynamics of smooth muscle cells, of actin in smooth muscle cells, lies a key transcription factor discovered in our lab many years ago known as myocardin. Myocardin is the master switch for smooth muscle cell differentiation, analogous to the role of MyoD in skeletal muscle. And so we've sought to further understand the molecular underpinnings of smooth muscle plasticity and differentiation, and to understand how this transcription factor is able to orchestrate the expression of smooth muscle genes, but also of cardiovascular genes, in which myocardin has been shown to play a pivotal role in reprogramming of fibroblasts to a cardiomyocyte cell fate. The myocardin protein is a massive one, 935 amino acids in length. It comes in 2 flavors due to splicing, shown here. And this protein, as we discovered subsequent to its initial discovery, represents the founding member of a larger family of transcriptional coactivators known as the MRTF family. And these proteins play myriad and essential roles in many aspects of cardiac, vascular, and skeletal muscle development, as well as development of various other cell types. And they contain a number of functional domains that enable them to execute these processes. Today, I'm going to focus on, in my final slides, just on one domain, and that is the C-terminal transcription activation domain. This is a pseudo-colored in situ hybridization, which I think really dramatically illustrates the specificity of expression of myocardin for the cardiovascular system. This is a mouse embryo in transverse and sagittal section, and you can see myocardin RNAs in red demarcating all smooth muscle cells of the aorta, the internal organs, and it also demarcates the early heart. And here you can see in a high magnification view of the aorta, the confinement of myocardin expression to the smooth muscle layer of this important blood vessel. The myocardin family, both myocardin and MRTFs, do not bind DNA directly, even though they are master transcription factors. Instead, they piggyback on top of a ubiquitous transcription factor called serum response factor, or SRF. SRF binds to this canonical sequence known as a card box, which fortuitously is located in the promoters or enhancers of most cardiovascular genes. When we first discovered myocardin, Daji Wang discovered this more than 20 years ago in our lab, we noted in the original paper that myocardin showed a fascinating punctate distribution in the nucleus, which at that time we did not understand the significance, but this image stood in the back of my mind for literally for decades, trying to think about what it might mean. And I'll tell you what it means in the remaining few slides. So this slide illustrates the intranuclear distribution of the myocardin partner SRF, which tethers myocardin to its target sequence, shown here. SRF by itself displays a diffuse intranuclear pattern, as you can see here in red. However, when SRF is expressed together with myocardin, as you saw in the previous slide, forms these nuclear condensates or puncti, and it draws SRF into those puncti such that it now goes from a diffuse distribution into a distribution identical to that of myocardin. And this is highly specific for SRF because another generic transcription factor, in this case NFYA, is not drawn into these nuclear puncti by myocardin. So we wanted to further dig into the molecular basis and functional significance of these nuclear puncta. To do so, we began by knocking in an EGFP reporter into the myocardin locus of human iPS cells, which we could then differentiate into smooth muscle cells or cardiomyocytes, and we could look at the intranuclear distribution of myocardin. So if we differentiate these human cells harboring the myocardin EGFP reporter into smooth muscle cells, you can see that myocardin adopts the intranuclear punctate pattern in these smooth muscle cells, marked by here ACTA2, the gene we just talked about in the previous portion of this talk. Or we can convert them into cardiomyocytes, and here those cardiomyocytes also display an intranuclear puncti or condensate distribution of myocardin, and you can see these cardiomyocytes are stained for cardiac troponin T. We've wanted to know what are the target genes of myocardin in each of these two cardiovascular cell lineages. And so by ChIP-seq, which was extremely enlightening, we found the following results. The top target sequence associated with myocardin, as you would expect, is a card box, the serum response factor target. So myocardin is drawn to SRF to these target genes in cardiomyocytes, as well as in smooth muscle cells. But there are many other interesting genes to which myocardin can be tethered. In cardiomyocytes, it is also associated with GATA factors, which are critical regulators of many aspects of cardiac muscle development. And very interestingly, in smooth muscle cells, myocardin is tethered not only to GATA factors, but also to TEAD factors, which are the downstream targets of the hippo signaling pathway, which has been shown to be a critical regulator of cell proliferation and pathogenic remodeling, as shown by James Martin and many others. And we're keenly interested in further digging into the functions and physiological significance of these various target genes. This is the structure of myocardium as I described to you earlier, and this is the C-terminal region, which is the transcription activation domain. If we delete the transcription activation domain from myocardium, it loses all of its ability to switch on smooth and cardiac muscle genes, even though it can still be tethered to SRF through this N-terminal region. Without the activation domain, it can't switch on these downstream genes. So we wondered, what is the physiologic significance of this activation domain with respect to the intranuclear condensate distribution of myocardium, and you can see that here. This shows high-resolution microscopy of myocardium forming puncti in the nucleus, and if we delete the activation domain, it still goes to the nucleus, but it adopts a diffuse pattern without puncti formation, and you can see that quantified here. So the activation domain is necessary for myocardium to form these intranuclear condensates. Is it sufficient? Well, I'll come to that in a moment. This is the transcription activation domain, which is localized to a 223 amino acid sequence at the extreme C terminus. And we noticed that this sequence was filled with aromatic amino acids, tyrosine, phenylalanine, and tryptophan, as shown here. And these hydrophobic aromatic residues have been shown in a subset of other proteins to be involved in condensate formation or the formation of puncti. So we wondered whether they might be responsible for the condensate formation of myocardium. And you can see that here. If we purify the myocardium activation domain by itself, fused to GFP, it actually forms droplets or condensates in solution. And if we change the aromatic residues to alanines in the activation domain, it completely loses the ability to form condensates. So clearly, there is a molecular basis for condensate formation that is mediated by these aromatic residues that are enriched in the activation domain. So what about the ability of myocardium to reprogram cells, in this case into smooth muscle cells? So this is a fibroblast reprogramming assay where we take fibroblasts and express eGFP myocardium, and then we look at whether it can reprogram them to smooth muscle. So in wild-type fibroblasts, myocardium forms puncti, and it switches on smooth muscle genes. Here's ACTA2 again, which you're already familiar with. And as I've told you, if you delete the activation domain, it no longer forms puncti, and it can't switch on smooth muscle genes. Or if we just change the aromatic residues in the full-length myocardium, again, it loses its reprogramming activity. And you can see that here. So the aromatic residues in the activation domain are required for condensate formation, and condensate formation is synonymous in this case with reprogramming to a smooth muscle fate. Now, many of you are aware that there have been a number of condensate-forming proteins that have been described in the literature, and we wondered whether all condensate-forming domains are created equal or whether there's specificity among them. And so we chopped off the condensate-forming domain from myocardium, as you've seen here, and that eliminates its reprogramming activity. And then we fused the remaining portion of myocardium to the condensate-forming domains of many other proteins, such as FUS, involved in RNA binding, TAF15, involved in TATA binding, DDX4, and even a receptor that forms condensates. When we put the condensate domain onto the otherwise inactive SRF binding domain, it can reprogram those cells. And I don't know why I've got red lines coming across my slide, but there's specificity with respect to how this works. And you can see if we take another condensate domain from an unrelated protein, it can't reprogram. Something is happening with my slides. Please overlook the red lines. I'm not sure if you can see them or not. So what I've told you then, and I'm just about to wrap up here, is that myocardium condensates are coupled to gene activation. The myocardium transcription activation domain enables condensate formation and lineage specification. And we think the condensate domains enable myocardium to exceed a threshold that can then condense components of the transcriptional machinery to switch on downstream targets. And through proteomics, which I haven't had time to talk to you about today, we've identified many components of these condensates that are involved in transcriptional specificity. Now, lastly, I've told you that myocardial activation domain confers upon it the ability to reprogram cells to a cardiovascular fate, whereas SRF provides the DNA binding specificity that brings this two-component system together. We wondered whether we could engineer a customized transcription factor that converts this system from a two-component system to a one-component system. And in doing so, we took the condensate domain of myocardial and we fused it to the C-terminus of SRF to create this chimeric protein. And when we did that, we found that this creates a super activator of cardiovascular gene expression. So in the left panel, you can see SRF, which you already know forms a diffuse pattern in the nucleus of cells. If we fuse myocardial condensate domain to SRF, now SRF adopts this condensate pattern in the nucleus and it switches on smooth muscle genes shown here by ACTA2. And if we mutate the aromatic residues in the condensate domain of myocardial in this context, it loses all of its ability to activate. So what I've told you then in closing is that SRF is a weak activator of transcription. It's devoid of condensate forming properties. In the presence of myocardial is recruited to SRF and it consolidates a transcriptional complex to switch on downstream smooth muscle genes. And this is a signal responsive mechanism, which I haven't had time to go into today. And then I've told you lastly that we can create using AI to basically predict the appropriate domains to fuse together, that we can create a super activator of cardiovascular gene expression by combining the condensate formation domain of myocardial with SRF, and this creates a super activator of cardiovascular gene expression. So this brings me to my last data slide, and it raises a number of really interesting questions that I think will keep us busy for many years. For example, do myocardial condensates control cardiovascular development in vivo? And what about disease? Are they involved in aspects of cardiovascular disease? Do MRTFs, the myocardial-related factors, which modulate many different types of phenotypes, use similar or different regulatory strategies? How is myocardial condensate assembly and disassembly regulated? And are there post-translational modifications, for example, phosphorylation or lactylation or others that might modulate condensate assembly? And how can condensate domains be further engineered, as I described to you in the final slides, to fine-tune transcription, perhaps by creating customized transcription factors? So I've told you today about two projects in my lab, the myoediting of muscle and cardiovascular disease and condensate control of vascular development. The muscle field has really provided fertile ground for exploration, and here I've summarized some of the many other projects that are being pursued in our lab by various trainees. And we fully expect that many more interesting discoveries will come from these areas of investigation in the future. And if there are any young trainees out there that are interested in this area, we're always happy to talk to you. And so lastly, this is my group. I haven't had time to talk about everyone's projects today. I'm sorry about that. But the people that were involved in this work are shown here. We had a DMD group that I talked about at the very beginning. We've had collaborations along the way with Luke Vandenberg's group and members of his team. Our LNP work has been carried out in collaboration with Daniel Seagord. I talked to you about the actin editing work and our work on myocardial condensate formation, which was initiated by Pei-Heng Gan, who has his own lab at Temple and has been in collaboration with Ben Sabari. We've had outstanding technical support in our lab as well as financial support from these organizations. And my long-term scientific partner who's been involved in everything and could have given this talk better than me is Dr. Ning Liu, who's shown here. And with that, I'm happy to stop. And if there's questions, I'm happy to take them. Thank you.